Chapter 12, we're going to cover blood. There are a variety of different body fluids found in the human body. Blood, which consists of blood cells and plasma. Plasma being the fluid portion of blood. Serum is when plasma is obtained minus the clotting factors, known as fibrinogen. And interstitial fluid. This is a fluid that's found between cells and tissues. Lymph is fluid found in lymphatic vessels. Blood is not really a body fluid like tears, mucus, saliva, or urine. It's a living tissue. It consists of living cells within a liquid matrix. Arterial blood is bright red. This is due to the presence of oxygenated hemoglobin, oxyhemoglobin. Venous blood is dark red, though it looks blue through your veins and skin. The function of blood is to transport oxygen from the lungs to your body cells and to return carbon dioxide from your body cells to your lungs. Blood also helps to regulate water content of your body. This is very important for water balance homeostasis. Blood helps to protect the body against diseases by the presence of antibodies. Platelets found in blood help to seal damaged blood vessels. The pH of blood is about 7.4. The average adult has about 5 liters of blood. It's a very viscous fluid. About 55% per volume of blood consists of plasma. This is the fluid portion of blood. Plasma is very clear, kind of a straw-colored fluid. Plasma does not contain any of the formed elements. Formed elements are the blood cells. It's a liquid solid, about 93% water, 7% solute. Plasma contains salt, ions, nutrients, hormones, proteins, clotting factors, antibodies, lipids, including your HDL and LDLs, which are lipid transporters, oxygen, carbon dioxide, and cellular waste such as urea. This slide shows a breakdown of blood and its constituents. Per volume, about 45% of blood is the red blood cells, white blood cells, and platelets. A hematocrit is a test that's done to determine the percentage of red blood cells in whole blood, the average range being 37 to 54%. If the range is less than 37%, that indicates anemia, and this may impair the blood's ability to carry enough oxygen to your body tissues. Hematopoiesis is the production of blood cells from stem cells found in the bone marrow. Erythropoietin, EPO, is a hormone released by the kidneys and liver in response to low blood oxygen levels. Erythropoietin is released in the bloodstream where it travels to the red bone marrow. It increases the number of red blood cells that are produced, increasing the oxygen carrying capacity. The levels of EPO will then decrease. There is a variety of dietary factors that are necessary to the production of red blood cells. Vitamin B12 and folic acid are necessary for DNA synthesis, which is required for replication of the cells within the bone marrow. Iron is needed to manufacture hemoglobin, the oxygen-binding protein found in red blood cells. Anemia is a deficiency in red blood cells, or the number of red blood cells is accurate, but the quality of the hemoglobin contained within the red blood cells is deficient. Red blood cells. Red blood cells are just a sac of hemoglobin. These are cells that don't have any other organelles. They look like biconcave discs, meaning they're wider on the edges, very thin in the middle. Sometimes in a light microscope, it almost looks as though you can see through them, like they're donuts. They contain about 1/3 hemoglobin. In arterial blood, erythrocytes appear bright red due to the presence of oxyhemoglobin. In venous blood, where some of the hemoglobin has been deoxygenated, the blood will look a little darker. Here you can see how narrow the red blood cells are in the center, the tops and the bottom. Red blood cell count. In males, average red blood cell count is between 4.7 and 6.1 million cells per millimeter cubed. In females, it's a little bit lower, about 4.2 to 5.4 cells per millimeter cubed. 5 million is a nice average number that kind of works for males and females. Red blood cells are very short-lived. They only live about 120 days. Red blood cells only live about 120 days. They are small enough to fit through the tiniest capillaries and become somewhat frayed and damaged. Macrophages in the liver and spleen phagocytize red blood cells that are old. What to do with the leftover hemoglobin? The hemoglobin from decomposed red blood cells is converted to heme and globin. The heme part is decomposed into iron and biliverdin, which is converted to bilirubin and excreted in the bile. The iron is reused to manufacture new red blood cells. The globin part, the protein part, contains 4 polypeptide chains, which can be broken down to amino acids and reused. The APR module has a really good video on hemoglobin breakdown. The small intestine absorbs nutrients. The blood transports these nutrients. These nutrients are used to manufacture red blood cells, which are produced in the red bone marrow. They circulate the blood for about 120 days. They become kind of old, fragmented, and macrophages break them down. Iron is recycled in the bloodstream, and biliverdin and bilirubin are excreted in the bile. White blood cells, or leukocytes, have an average value of about 10,000 per millimeter cubed. There's a variety of different categories of white blood cells, and all have a role in fighting disease or inflammation. Some actually contribute to the formation of allergies. The main function of white blood cells is protection of the body as part of the immune system. White blood cells attack and destroy bacteria and pathogens. They also remove dead cells and tissues. Diapedesis is a method in which white blood cells are able to exit blood capillaries and travel to the site of infection. Let's look at the classification of white blood cells. White blood cells are classified in 2 groups based on the appearance or absence of cytoplasmic granules. The granular sites have cytoplasmic granules. This includes the neutrophils, eosinophils, and basophils. There are 2 groups of white blood cells that don't have cytoplasmic granules. These are the monocytes and lymphocytes. The neutrophils are the most prevalent white blood cells. They function in phagocytosis. They're able to attack foreign debris or substances. The eosinophils are active in allergic reactions or parasitic infections. Basophils release histamine. Histamine increases capillary permeability, allowing fluids in white blood cells to access an area of infection. If this occurs when you're having an allergic reaction or having an allergy, you tend to take an antihistamine. Lymphocytes provide specific immunity. B cells, which mature in your bone marrow, make antibodies. T cells, which mature in your thymus, consist of a number of different types. One type, the cytotoxic T cells, help to target potentially cancerous cells. Monocytes can also function in phagocytosis. This is a normal blood smear. In a normal blood smear, you would see a prevalence of red blood cells, since there's 5 million per millimeter cubed, and 1 or 2 white blood cells. The white blood cells present here are a neutrophil on the left and a lymphocyte on the right. The easiest way to remember the prevalence of leukocytes is with this little acronym: never let monkeys eat bananas. Most prevalent leukocyte is neutrophil, followed by lymphocyte, monocyte, eosinophil, and then basophil. There's many different types of leukemia. Leukemia is when you have an abnormal proliferation of white blood cells, abnormal types, abnormal forms, and numbers. On the left is a normal blood smear. On the right is a specimen with leukemia. You can see that there's lymphocytes, but they have a very abnormal shape. Some of them almost look rectangular. Platelets are involved in blood clotting. They're also known as thrombocytes. Normal levels 150,000 to 350,000 per millimeter cubed. These are actually very small cell fragments produced by megakaryocytes, very large cells that are found within the bone marrow. Thrombocytes have a very short lifespan of only about 1 to 2 weeks. Hemostasis means a stoppage of bleeding, and this occurs in 3 steps. First, the blood vessel spasms, then platelets form a plug, and finally blood coagulation occurs. Here we see a vascular injury. The first response is for the vessel to constrict and for surrounding tissues to push inward. This will help slow down blood loss. The platelets, which are flowing down the smooth endothelial lining, detect the foreign collagen and start to adhere together. This forms a platelet plug. The platelet plug helps to control blood loss. The formation of a blood clot is a cascade reaction. There's a variety of proteins found in the bloodstream, and ultimately fibrinogen is converted to fibrin. Think of fibrin like fiber threads, and this reinforces the platelet plug, forming a blood clot. The reason why this is so carefully regulated is you want blood clots to be able to form quickly when there's vascular injury, but you don't want blood clots to form when they're not needed. This slide shows a blood clot. A thrombus is an abnormally formed blood clot. Sometimes these occur in the deep veins within the legs. If a person is sitting for a very long period of time, blood pooling can contribute to the formation of an abnormal blood clot. This becomes particularly dangerous if the thrombus moves. This is called an embolus. And if the embolus travels to the lungs, it can cause a pulmonary embolism and potentially death. Blood groups and transfusions. The clumping of red blood cells following an incompatible transfusion is called agglutination. This is due to the interactions of proteins on the surface of red blood cells, known as antigens, with antibodies that are carried in the plasma. Let's look at the ABO blood group. There are other antigen groups found on red blood cells, but it's the ABO group that's critical for giving a proper transfusion. Type A blood. People with type A blood have A antigens on their red blood cells. They would sense B antigens as foreign and therefore have anti-B antibodies in their plasma. People with type B blood have B antigens on the red blood cells and type A antibodies in their plasma, right? Since if you're type B, you would sense type A as being foreign. People that are type AB have both types of antigens and therefore do not have antibodies to either. They're referred to as a universal recipient. In theory, they could receive any of the other types of blood. When given a transfusion, though, typically you try to give somebody the exact same type blood. Type O blood doesn't have either of the antigens. However, they have antibodies to both type A and type B antigen. They're referred to as a universal donor. When given a transfusion of blood, you're not concerned with the antibodies in the plasma because transfusions are given with packed cells. You're concerned about the antigens that are present on the red blood cells that are being transfused. This just gives you a visual representation of what like an A antigen, B antigen, AB antigen, or absence of antigens would look like. On the left is a normal blood slide. On the right is blood that has agglutinated. There's an additional factor, sometimes called antigen D, that's found on the surface of red blood cells. And another name for it is the Rh factor. This name originates from where it was first discovered in rhesus monkeys. This is an antigen that either you have or you don't have. If you have this antigen D, you're considered to be Rh positive. If you don't have this antigen, you're Rh negative. This is a type of immunity that's acquired, so individuals that are Rh negative do not have any antibodies to Rh positive factor, unless they've been exposed to that factor once. Rh negative individuals, once exposed to Rh factor, will then start to produce the antibodies to target the Rh positive antigen. This is of concern when you have an Rh negative woman who carries an Rh positive baby. Sometime during delivery, there's a mixing of blood and she's now been exposed to the Rh positive antigen. She will now start to make antibodies against the Rh factor that could attack the blood cells of the second Rh positive baby. This is called erythroblastosis fetalis. However, there is a drug that was manufactured that desensitizes these antibodies and prevents this from occurring. Try the link below to do the blood typing game. Try to save some of the patients' lives and you can learn more about human blood types. There's a variety of Greek and Latin words associated with blood, such as erythro, meaning red, hema, hemo, blood, leuko, white, osis, means abnormal condition. When used with words related to blood, it means abnormal increase. Poiesis, to make or produce. Stasis we did previously, stop or stand still. And throb means clot. Red blood cells, also known as erythrocytes. Stoppage of bleeding, hemostasis. Blood clot cell, thrombocyte, normally known as platelet. An elevated white blood cell count is known as leukocytosis, and the formation of red blood cells, erythropoiesis. That concludes Chapter 12.